Good evening, everybody. Inaugural lectures are one of the highlights of the academic year, and we're in for a treat this evening, I'm sure. So I'm Gary Taylor, I'm the Deputy Principal, and I'm here to introduce the Inaugural, which tonight is being given by Professor Andy Lynch. So Andy studied mathematics at Cambridge and then stayed on a year to do advanced studies in mathematics, where he largely focused on statistics. A PhD at the University of Sheffield followed, and that was sponsored by the Horticultural Research International and was centred on methods to improve early season prediction of crop yields. He then went on to do a postdoc at the University of Newcastle for a year before returning to Cambridge in 2002, where he took up a position as a research associate in the Centre for Applied Medical Statistics, Public Health and Primary Care at Addenbrooke's Hospital. And there he was a consulting statistician on over 100 research projects, including genomic studies, which led to an interest in cancer genomics. From 2006, he worked in that field in Cambridge, first at the Department of Oncology, and then the Cancer Research UK Cambridge Institute, with whom he still maintains links. Happily for us, he was appointed as Professor of Statistics in Bioscience at St Andrews in June last year. And this is a joint appointment between the School of Medicine and the School of Mathematics and Statistics. His research is a mixture of analytical method development and application to clinical cancer projects. And he has worked on projects related to ovarian, breast, esophageal and brain cancers, but his main focus at present is prostate cancer. And he is co-principal investigator on the CRUK-funded International Cancer Genome Consortium, the prostate cancer study. Now, the science of genomics is one of the great scientific revolutions of the 21st century, and you may recall back in the year 2000, a remarkable joint transatlantic press conference given by President Clinton and Prime Minister Blair when the preliminary human genome sequence was published. The project was finally completed in 2003, and this project had taken 15 years, involved a collaboration of 6 countries and cost $2.7 billion. To sequence all 3 billion bases of the human genome. 10 years ago, the cost had come down to $10 million, and today it costs about $1,000. And this revolution in the acquisition and analysis of big data sets requires cutting-edge statistical methods. And at $1,000 a genome, the hope of personalized medicine is truly a reality. And so tonight, we're very privileged to have Andy Lynch give us a glimpse into this exciting science with his inaugural lecture entitled Getting Cancer's Number, an Interface of Statistics and Molecular Biology. Professor Lynch.
Well, thank you, everyone, for coming, despite the weather we've had today. I assume you're all here because you have an interest in cancer research. If you don't have an interest in cancer research, I'm told that the wine won't arrive any earlier if I stop talking, so you may as well just put up with me. I'm going to tell a few stories today, and I hope that everyone will encounter something they haven't seen before, and hopefully something that will interest them. But before I talk about cancer research, as promised, I just want to introduce a paper, or rather a study, that was my first glimpse into this world and has influenced how I've seen everything since. And this was, as I say, not in cancer research, not even in biology, this is designing an electrical circuit. And the aim of this circuit is to distinguish between 2 audio inputs, so a high-pitched frequency and a low-pitched frequency. Now, this is being implemented on what's called a field programmable gate array, and I know nothing of electrical things and very little of FPGAs, so I can't tell you much more than that. But this is an array of 100 cells in a 10 by 10 grid. Each of these cells has 18 settings that can be on or off, and you configure a cell to being one of several logic gates by setting those 18 on-off switches. And this can be done automatically by computer, so this doesn't take much time at all. Despite that, with 100 cells and 18 options in each one, we end up with 1,800 options to set, each of which can be on or off. So there are 2 to the 1,800 potential designs. As quick as it is to set one of these things with a computer, you cannot explore a number that large just by trying each one in turn. So what Adrian Thompson did was to attempt to arrive at the solution via evolution. So all you need for evolution is a rule that tells you what your fitness is, so how well you're doing compared to your objective, and a rule for breeding 2 of your circuits. I will leave that aspect to your imaginations. But if you have such a rule, if you have such a rule, then you can set these things breeding, and starting off with a random population of circuits that doesn't do very well at all, by the time you get to 3,000 generations of circuits, the best in the population is actually performing the job almost perfectly. Well, that's what evolution is meant to do. That's not in itself a surprise. But there are several things that are surprising about the solution it comes up with. The first thing is actually that it works at all, given that distinguishing frequencies is essentially a question of timing, and there is no timing facility on this gate array. The second thing that's worthy of note is the fact that despite there being no penalty for the circuit, despite there being no penalty for the circuit to take up space, it's actually confined itself to a solution that only occupies perhaps a third of the array. The really remarkable thing was that whilst there's a circuit of logical gates that goes all around here and eventually comes out of the output, there are 5 cells that are shaded here that are not being used as part of a logical circuit, but if you change any one of them, this ceases to function. So evolution has found a secondary use, not beyond the primary functions, of these cells. And perhaps because of that, unsurprisingly, if you try and apply this solution to a theoretically identical gate array, it will not work. Well, 2 to the 1,800 is quite a large number, but the DNA you inherit from one parent is a code with 2 to the 6 billion potential sequences. And we've been evolving for a while, so many of those are also now fixed in place. But still, between any 2 people, there are millions of ways that we vary. And cancer is very much a disease of mutations of that genome. And understanding those large data sets and those sequences and how they arrive is key to making headway and understanding how we can approach cancer. And so the sorts of questions we're going to be looking to ask, ideally, would be what goes wrong in a cell that causes it to become cancerous? When did this happen? If we know that, could we do something to prevent it happening? And failing that, can we do something after the event to halt the cancer in its tracks? I'll pretty much only be able to talk about the first of those this evening, but I will at least get on to mentioning some treatment options that arise out of it in passing. Now, if you happen to come to Professor Chaplin's talk in April, you will have seen a very similar picture to this, although it's a generational thing. I think Mark used the original Hanahan and Weinberg picture, whereas this is the Hallmarks of Cancer, the Next Generation, which came out in 2011. What Hanahan and Weinberg depict here are hallmarks and characteristics of cancer that cancers need to have acquired in order for them to be successful cancers. And so these are things like obtaining a blood supply, avoiding the immune system, and so forth. And the 2 that I will be talking about today in any detail are the fact that there is an inherent limit on how many times a cell can divide, usually, which cancers have to overcome. And typically, cancers exhibit a great deal of genome instability, leading to a lot of mutations that can enact these other characteristics. And we will look at that in great detail. Now, those mutations can happen on many scales. So we could have entire genomes being duplicated in a cell, perhaps due to cell division going wrong. So rather than having 46 chromosomes in a cell, we end up with 92. Or these things can happen at the chromosomal level. So just a particular chromosome, you should have 2 copies, but you might end up with 4, 5, or perhaps only 1. Intrachromosomal breaks can occur and fusions can occur, so you might end up with, for instance, part of chromosome 5 attached to chromosome 3. You might see regions being replicated several times in tandem, for example. You can have insertions or deletions of DNA from one base, one letter, up to millions. And what we're going to focus on for the most part of today, changes to individual bases. So in your sequence of letters, where you've got an alphabet of A, C, G, and T, one letter has changed in one position. Now, how we go about identifying these is conceptually quite simple. I'm aware that I have several of my colleagues from statistics here who don't tend to work in the cancer domain. Many of them work more in ecological statistics. So I've tried to come up with an example that will be more welcoming to you. So essentially what we have to do is we have to take the DNA of a cancer and sequence it, and DNA of matching tissue from the same individual that isn't cancerous and sequence that, and then we play spot the difference. Go on. Sorry? The crab, yes, that's how we know this one's cancer. Any more? Top left, yeah. Quasi's missing there.
There's a green fish on the right.
The octopus, yes. Yeah, and something missing there. Right. So we can take our cancer and our benign tissue, we can identify all the mutations we can, we list them. We could at this point try and bring in our biological knowledge of the system and say, well, actually I can prioritize some of these mutations because due to my expertise in this field, I know that the absence of Lieutenant Quasi-Cat is going to have more impact on this system than the crab appearing. And we can do that. But when you try to do that in real life in a cancer setting, first of all, you don't just have 4 mutations, you have several thousand, typically. But also, it turns out 2 labs do not agree on the results. So we took part in a study as part of ICGC that the master mentioned, where the same data set was sent out to many labs that do this around the world, including ours, and we all had to call the mutations and then send our results back. And this is showing the performance of all different labs, so each blue square is a lab that sent in their results. And so you've got some over here that just wanted to find all the mutations they could, and they didn't care, apparently, if they called things as mutations that weren't, as long as they picked up every mutation that was available to them. You've got some here, or one here rather, who just called a few mutations, but when they call a mutation, by goodness, you can tell that that's really a mutation. Then you've got ones up here who are sort of all-rounders, trying to balance the two. And depending on how you're going to use these data, you can pretty much justify being anywhere on this front of all these labs. And these ones as well, because of idiosyncrasies of sequencing and differences between cancer types, probably these labs, if they were looking at their own data from a cancer that they're used to looking at, would also be working at that front. So people do okay, but as you can see, there's quite a bit of difference as to what that actual list of mutations should be. And I should credit Matt Eldridge, who actually did most of the work for our group's entry into this, not competition, comradely exploration of science. Yes. So that's skipping ahead to whole genome sequencing, and I want to bring you back to where I entered this field. So I was working as a medical statistician at Addenbrooke's Hospital, and Ahmed and colleagues arrived with a question about TP53 mutations in ovarian cancer. And again, if you were here in April for Professor Chaplin's talk, you will have heard something about TP53. TP53 is the most commonly mutated gene if you look across a whole range of cancer types. And it has local connections because it was discovered and partly characterized in Dundee. Yes, my artistic skills are lacking, I'm sorry. It's also a reminder that this isn't necessarily just a genomic condition, because the HPV virus, which is responsible for so many cervical cancers and other cancers, acts by, partly at least, by interfering with the TP53 protein. And then one of the quirkier headlines in cancer news headlines in recent years was the revelation that one of the explanations for the low cancer rates in elephants is the fact that they have 20 copies of this TP53 gene compared to our single copy. And this is presumed to be an influence on their lack of cancer rates. But Ahmed did not come to me with questions about elephants. He came wanting to know, of the papers that have been published recently, some claimed that serious ovarian cancers that had a TP53 mutation had a worse prognosis than those that didn't have that mutation. And those papers are the black dots on this figure. Conversely, there had been several publications that said there was no prognostic value to identifying this TP53 mutation. And these are the open squares. Now, and we were embarking on a systematic review which identified these as being the vaguely reliable studies to include, and we were going to try and answer the question, is there a prognostic value to this mutation or not? And you might look at this immediately and think, well, the white squares are outnumbered by the black circles, but actually, although all these black circles say there's a prognostic value to having a TP53 mutation, they do not necessarily agree on what that is or in what context it arises. So they shouldn't be regarded as agreeing with each other. And while we were trying to dissect what was going on here, we couldn't help noticing that there was a very large range reported in the number of cases of ovarian cancers that have a TP53 mutation, ranging from about 25% to 75%. And this we couldn't explain because all of these studies have been chosen to be, broadly speaking, population studies. We noticed, though, that when we ordered them by the year in which they were published, with the exception of one more recent paper that ruined everything, there seemed to be a trend that the more recent paper was, the more cases seemed to have a TP53 mutation. And in fact, when we ordered the papers by the method used and how convincing that was to identify a TP53 mutation, this became almost linear. So some papers were using really quite indirect ways of inferring that there was a TP53 mutation, and they were being quite open about this and saying that they were underreporting. So we thought the truth was probably that actually the true rate would be somewhere around 75 to 80% or more. And the upshot of this was we didn't publish a systematic review, but rather Ahmed and colleagues went away and performed their own study to get a handle on what this rate was and whether it has a prognostic value. And they recruited more collaborators, and it turns out that there is no prognostic value to simply having a TP53 mutation in high-grade ovarian cancer, because every cancer has one, to within a waving of a hand. So, well, that put paid to that as a possibility, but this opens up other possibilities. The fact that every serious ovarian cancer has a TP53 mutation gives us something to work on in terms of a biomarker. If you've treated a woman and now you're trying to monitor whether the cancer has recurred, you can look for a very small part of the genome in circulating DNA in the blood and check whether the cancer has returned. And this is active work of James and I think probably several other people in this original study. But this was my brief flirtation with ovarian cancer. I haven't worked with it since. But that's one gene. That's one gene out of 20,000, and those 20,000 make up about 2% of the genome. What do we do if we want to look across the entire genome? Well, as has been mentioned, sequencing a genome is now a lot easier than it used to be. But there are a few things you should know. This is the end of the Octonauts theme, I promise. Sequencing a genome is now like doing a jigsaw puzzle. So we don't put DNA into a machine and get 6 billion bases in sequence coming out of it. Rather, we put little bits of DNA into the machine and get little strings of readings of DNA out of it, and then we have to try and piece them together like a puzzle. And when you're doing a puzzle, you've got several things working for you. You've got the shape of the pieces. You've got the fact that some details of the picture go from one piece to its neighbour. And of course, you've got the picture on the box. All these things are helping you. When we sequence a genome, we don't have these. Our picture would look something more like this. So this would be a very small genome, but I couldn't get it to go any larger than that and still be visible. We don't have nicely shaped pieces like this. We just have uniform pieces, and we have no features that go from one piece to the next. So how do we approach such a puzzle? Well, the good news is that the pieces that we generate will overlap. So typically, one of our genome puzzles will have 500 million overlapping pieces, each of which would cover about 1/10 millionth of the genome. And so you can work out from that on average, any part of the genome should have 50 pieces roughly covering it. And because these pieces overlap, and they're large enough that we can identify those overlaps, we could start to build up larger and larger pieces by seeing that this piece overlaps with what's there, so we can put it down, and that would fill in 2 more squares. Well, that's one approach. But the other thing is that we've got a picture on the box, and that's what the Human Genome Project gave us. And it's fashionable to say that the Human Genome Project did took 15 years and cost $3 billion, and we could now sequence 5 before breakfast on a Tuesday if we want to. But that ignores the fact that we can only do it partly, yes, because of new technologies, but partly because we've got the picture on the box. Otherwise, the computational complexity would be much greater, and the computational costs of this sort of thing probably now outweigh the costs of actually producing the sequences. We do, though, have to recognize that there are some problems. That picture on the box is for a generic genome. It is not for the genome that we've sequenced. It's also the case that the sequencer makes errors. So every now and then, one of your pieces will have the wrong color. We also don't actually have even coverage. Now, you know how annoying it is when you buy a jigsaw and there's a piece missing. Well, due to biases and due to just random sampling, some bits of the genome, when we do the sequencing, we do not cover. And finally, there are parts of the genome that are just repetitive, so you've got the same picture appearing in 2 places, and mapping back to them is tricky. An extreme case in point there are the telomeres. Now, this is Henry. Henry was my student. We spent 4 years working on looking at telomeres from this sort of data. And he's Dr. Farmery, so it was successful. This is good news. Now, the telomeres lie at the ends of chromosomes. And they, to some extent, provide protection for chromosomes, because without them, you have a lot of instability, inducing mutations, which would be bad for reasons that sort of intuitively obvious. You do not want mutations in your cells, if possible. But they also provide the limit on how often a cell can reproduce. Because every time you copy a chromosome, your copy is a little bit shorter. You miss a little bit off the end. And because the telomeres are at the end, what you miss is a little bit of telomere. So as your cells divide and reproduce, your telomeres are getting shorter, and at some point they will head to a point where they cause catastrophic genomic instability, and you will get all sorts of mutations occurring. But before that point, the cell should recognize that they've reached a limit, and it will stop dividing. The cliche, when talking about telomeres, is to describe them as being the aglets on shoelaces. Now, I prefer to think of them as the leader tape on cassettes. There are two reasons for this. One, I have never tried to store data on a shoelace, although I will conceive that with weaving it's possible. I also have never tried to copy a shoelace. The downside of the tape analogy is that you do now have to spend a lot of time explaining to some people what tapes are. As I say, telomeres place a limit on how often cells can divide, how often they can reproduce. That's all well and good, except you have some cells that you do want to reproduce indefinitely. So your stem cells, and in men, the germ cells, the sperm. And one of the consequences of this is this quirky fact that if you're an older father, if you're older when you have a child, you will pass on to them longer telomeres, because the telomerase, which is a mechanism that cells can use to extend telomere length, has been active in the male germ cells for all of your life. And this paper in 2012 showed that actually you can detect this in the grandfather's age at the time of a child's birth as well. Now, there are statisticians in the audience. This is a good paper, but a terrible figure. So this scale makes it look as though there's a huge difference between this box and this box, whereas, of course, that's slightly artificial. However, there is a definite change, and it's seen not only in this paper, but in others as well. And just as an aside, it's worth thinking about the fact that over the last 50 years, the average age of fathers in the UK has been getting older. So presumably, we've got a population that's now got longer telomeres. What implications that has for health, I do not know. So this is the limit on cells reproducing, and cancer cells have to overcome this limit, and they can use the same method that stem cells do. They can employ the protein telomerase, or they can employ an alternative lengthening of telomeres method, which is referred to as alternative lengthening of telomeres, or ALT. Clearly not discovered by fly biologists, or it would have a funkier name. But there's a sort of tension in timing here, because your cell also wants to gain lots of mutations and exhibit genomic instability if it's going to be a successful cancer. So the timing of when this kicks in is going to be crucial for the nature of the cancer that develops. Okay. We don't get to see the telomeres at any time other than when the cancer is mature, however, but it's still worth, and it's still been beneficial, looking at them in several cancer types. So going back to the jigsaw analogy, which I'm going to stretch as far as I can, what we find is that this white area represents the telomeres, and we're trying to estimate this telomere length here. Now, our problem is that these jigsaw pieces are just completely white. They could go anywhere. What we could do is just count up how many we've got, and then we could estimate the area here. And if we knew the width, the height, then, you know, my ears of mathematics tells me that if I know the area of a rectangle and I know the height, I can estimate the width. However, that works in non-cancerous cells where we know the number of telomeres that are present. It doesn't work in cancer cells where we don't know the number of telomeres. And much of what Henry did for his PhD thesis was devising ways of estimating this width when we don't know what this height is. And, you know, it's not quite as easy as it looks in the jigsaw analogy, I have to say. So, I just got time for this. Okay, this is an interlude, but this is the most astonishing result I've ever seen from genome sequencing, and I wanted to share it with you. And to get to this result, we just need to be clear on one thing, and that is when a child inherits 2 chromosomes, one paternally, one maternally, and then passes a chromosome on to its offspring, it doesn't pass on one or other of the ones it's inherited, it passes on a mixture. And so if you think now of 4 synthetic organisms that we've generated in the lab or in silico on the computer, if we generated 4 completely independent individuals and then mated them in this manner, you would find a grandchild here where this copy of the molecule contains material from these 2 grandparents, this copy of the chromosome contains material from these 2 grandparents, and if we drew a map and colored in black anywhere where the chromosome had the same material in both copies, it would be blank. If, however, we were to take 2 individuals initially, they had 2 children, and then those children mate, and this is where it's really important we're talking about synthetic organisms. You would find that this offspring would have chromosomes that are a mixture of all 4 chromosomes in the original generation, in both cases, and when you draw this map of where they have the same material in both copies, about a quarter of it would be coloured in now. And if we got really freaky and decided we're going to take 2 germ cells from this individual and force them to become a new individual, then both of these chromosomes would just be a mix of these 2, and an awful lot more of this map is now coloured in. And the principle here is that by drawing this map, you're learning something about how many individuals there were in the recent ancestry of this offspring. And what the Durbin lab in Cambridge did, which I find absolutely fascinating, is they took a human genome and going back over 1 million years, they estimated the size of the population of that individual's ancestry. And they did this for 6 individuals. They did it for 2 African individuals, 2 European, 2 East Asian. And let's see, somewhere around here, we emerge as a species. This is how far back we're going. I think that's right. And then all the way around here, all of our individuals have the same ancestry, locked into a region of Africa. And then at this point, they diverge, and it's probably not clear, but it's the 2 African individuals have an ancestry, a population that's larger than the Europeans and Asians who are now moving away. And when they get to the actual out of Africa migration event, there's a huge bottleneck in the population size, which can be seen here. And to me, the idea that you can take an individual's genome, albeit using a lot of knowledge gleaned from many other genomes that you've looked at, and trace back something that's a reasonable history of the human race. Astonishing was the word I used at the start. It's the word I'm sticking with. Okay. I need to speed up. Right. So the work I've been doing most of all in recent times has been on the International Cancer Genome Consortium, or ICGC. And one of the aims of this is to identify important cancer genes. Now, ICGC started off with the aim of getting genomes for 500 cases with matched tissue from 50 types of cancer and accumulating it in a uniform manner where scientists all around the world would have access to the data and we'd be able to make inroads into cancer research. As of spring this year, there are 84 projects worldwide contributing, looking at 22 cancer sites, but you can have multiple cancer types at a given site. And they nearly got to their initial target of 25,000 donors. And on average, there are about 4,000 mutations per cancer. Now, the two studies I've been involved in are the esophageal and prostate studies in the UK, and these days mainly the prostate one. And that's what I'm going to tell you about. So, prostate cancer is the leading cause of cancer death in men in the UK, with a little more than, over 10,000 a year. Oh, that should be 2015, I think. Never mind. But there are just under 50,000 cases every year, new cases every year. So you can see immediately that survival, as cancers go, is quite good here. Compared to the ovarian cancer, which has about 4,000 UK deaths a year, and a survival rate of 35% at 10 years, prostate has 84% survival at 10 years. So there's a lot of potential here for over-treating people. And this effort was, the UK branch of this prostate effort is initially to look at 250 cases with a matched benign tissue, looking at the genome sequence, but also lots of other data types that I won't tell you about today. Well, actually, I'll tell you about one today. And this is a collaboration with lots of sites around the UK and indeed Finland. Now, we've had a reasonable amount of success as a study. We've improved our understanding of how the tumours within a single prostate, or sorry, what might appear to be one tumour within a single prostate, has actually risen several times, apparently independently from a genomic point of view. We've improved models of how prostate cancer metastases spread around the body. We've improved our understanding of what's going on with the epigenetics of prostate cancer, and I'll talk about what that means later. We've had some results about metabolism, but probably the key one is our most recent paper, where we identified several new lines of potential treatment for prostate cancer based on the genomic results that came out of studying a large number. And the aims of the project that remain unfulfilled are, we'd really like to have a way of distinguishing advanced disease that requires immediate treatment from indolent disease, which probably would be best left alone. We want to understand the differences that we see in prostate cancer with ethnicity. So, as a white male, I have about a 1 in 8 lifetime chance of getting prostate cancer. If I were black, that would be 1 in 4. If I were Asian, it would be 1 in 12. There are various hypotheses as to why this is, but nothing conclusive. And that impacts our ability to target treatments. We'd like to know, at a molecular level, why these things are arising and why one prostate cancer differs to the next. We'd like to know that just out of scientific curiosity. Actually, even if we don't learn that, we could still identify subgroups of disease that would be clinically useful for targeting treatments. But we will do a much better job if we actually understand why these are different subgroups. Now, prostate cancer is very different to ovarian cancer. Ovarian cancer had the TP53 mutation, which is there in nearly 100% of all cases. In prostate cancer, the most commonly mutated gene is SPOP, which is only there, mutated in 11% of cases. The next most common, FOXA1, down to 3 cases. The next most common is IDH1 in 1%. And this is based on a study of 333 cases, not ours. If you look at all the prostate samples in ICGC, TP53 does actually come top again at 7%, but this is because TP53 is common in very advanced and metastatic disease in prostate cancer, and there are several of those in this population in the ICGC study at the moment. So, if we want to find more driver genes, which presumably are present, by a driver gene I mean one of these genes that's actually doing something, one of these genes that when mutated is actually driving prostate cancer, it's presumably there at most in 1% of cases, and we're going to need large data sets to actually find them. And so for this reason, amongst others, the Pan-Prostate Initiative we're trying to set up, where all the various prostate sequencing studies around the world, along with some other centres such as South Africa, which has collected material but not sequenced it for a number of African countries, and we're talking to various other Asian countries as well, trying to put together a large number of prostate genomes that would allow us to be able to identify those mutations that are really exceedingly rare but still driving disease. So that's one approach we can take. There are, however, alternatives that we can look at. So the first thing we could try to do is a pathway analysis. Now, this is a figure I've stolen from this paper showing the Hess genes and how they interact. I have to say, this work is now with Dr. Massey. Dr. Massey and I have also published a map of the Hess gene network. I have to accept that this one is prettier, so I'm showing this to you today. I do not necessarily agree with it all, but it will do for our purposes now. So what we can do is we can say, well, we know these genes are highly connected, so perhaps we don't need one gene to be mutated repeatedly. Perhaps it's enough that in 1% of cancers or 2% of cancers, one of these genes is mutated. And we can bring in this biological knowledge, bring in our data, and see if that happens. We could also start looking at the mutations that aren't in genes. And we do that to an extent, but they're so much harder to understand. And so we don't do as good a job yet. There are still lots of low-hanging fruit in genes that we're able to pick up. So even if we discover something that isn't in a gene that is apparently driving cancer, how we would go about treating it is not clear at this point in time. I mentioned at the front of the talk that there are other types of mutation, not just these point mutations. And prostate cancer does have a genomic rearrangement that's quite common, 40 to 50% of cases. However, recent results have cast doubt on whether it can be an early event in prostate cancer, and in any case, it still leaves 50% or 40% unanswered, unexplained. So that brings me to methylation. So methylation of DNA is not a change to the sequence of A's, C's, G's, and T's, but rather it's the addition of a methyl group to the DNA in the position of a C when it typically next to a G. And we've profiled that in some of our cases as well as the genomic sequence. And when we did this, we were very excited. Because if you look at the Hes5 gene, it shows a a clear change from benign tissue to tumor tissue in prostates. And it does that if you look across all the different sites in a prostate tumor that are otherwise genomically distinct. It does that if you look across a cohort of prostate cancers. It does that if you look across the entire region where we think methylation will control expression of the Hes5 gene. And we're particularly excited, not only because we found something that's more common in prostate cancers than anything else we've ever seen, but a colleague of ours, Al Lam, had shown that Hes6 is a driver of advanced prostate cancer. And this network wasn't chosen at random. You can see that Hes5 and Hes6 are very closely related. So we thought this is fantastic. We think that we're changing the region that controls the expression of the Hes5 gene. And so when we looked at comparing benign and prostate cancer at the expression of Hes5, we discovered there was absolutely no change. And that was slightly disappointing. But some digging and some lab work to fill in sort of conditions that we don't see in real life has led sort of leads me to suggest that what's happening is in the prostate cancer, we're switching from regulation of Hes5 being controlled by Hes6 to Hes5 being turned off, being controlled by methylation. And actually, it seems really important to the cancer that Hes5 is silenced in this manner. So that gives us something to work on. Okay. Excuse me. So I just want to come back to this plot. I mentioned earlier that each prostate cancer, on average, about 4,000 mutations. And in 15% of cases, we have one mutation that we can explain as driving the driving disease. So we have a lot of mutations that we aren't treating as drivers. We refer to them as passengers. And what can we do with those? Well, one thing we can do, if we classify those 4,000 or so mutations into 96 different mutation types, is say something about the processes that drove the mutations in that cancer. So this is, I was told I had to have an equation in this. So 4 times 6 times 4 is 96. I think that's there we go. So what we have, our 96 classes come from 6 possible mutations. You might think, hold on, there could be a G turning into a T, but due to the complementarity of the 2 strands of DNA, a G turning into a T is the same as a C turning into an A. And then before those 6 mutations, there are 4 potential bases, and afterwards there are 4 potential bases. And you can classify each of your mutations. You can create a table with 96 cells, or you can create a bar chart with 96 bars, and you can plot how they look. And this is being done in the style of work that came out of the Wellcome Trust Sanger Centre, where they also have identified the signatures of certain processes. So this is a process that just occurs naturally via aging. This is what mutations look like if they occur from tobacco, this from UV light. And in particular, so we can take the mutation pattern we see and we can try and build it up out of components that exist in this catalogue hosted at the Sanger. And in particular, we can sometimes separate out in our cancer the early mutations from the older mutation, the early mutations from the recent mutations. And we can start to see the changing nature of the factors that have influenced the mutations over time. Okay. Well, that's me done telling stories. I did promise in the abstract that I would mention some statistical challenges. I hope that as we've gone through, I've made it clear that quite a lot of these steps throw up many statistical challenges, but those are ones that are being dealt with by lots of groups around the world. Where I think we need to be doing more as statisticians is on the propagation and quantification of uncertainty, which is sort of our day job in these analyses. So for very good reasons, the analysis of sequencing some DNA, assembling that jigsaw, identifying the mutations, doing something downstream with those mutations, such as signature analysis, are done in a compartmentalized fashion in different labs possibly. And the uncertainty arising from each step doesn't necessarily get passed on to the next one in a way it should do. And then when you look at things like the pathway analysis I mentioned, where we're taking inferences from our data and combining them with prior biological knowledge, we can account for the errors, the uncertainty in our inferences from the data, but we tend to treat this as being true, when actually there's uncertainty on the biological knowledge we're importing as well. The second big challenge that really faces us at the moment is how we go about marrying lots of different data sets. Currently, what we're doing a lot of the time is just going back to the raw data and reanalyzing them. This is very expensive computationally, and in medical statistics and other branches of statistics as well, doing a meta-analysis on the results that have arisen from many studies is just a perfectly natural thing to do. We haven't yet got to the stage in genomics where we're able to do this satisfactorily. And there's also aspects of trying to integrate clinical patient data, such as ICGC, with data coming from cell line experiments in labs in a sensible manner, and working out how the uncertainties should be expressed in any results coming from that. And then the final challenge I think we have that isn't being addressed is assessing the complexity of our solutions when we come up with models. So quite often we call upon Occam's razor, the principle of Occam's razor, that the simplest solution is the best one to go with. And that's fine. That's fine. but I'm just not convinced we're actually assessing which is the simplest solution all the time. And if you think back to this synthetic biology, synthetic evolution question, we know that this is a simple solution because we saw the process that led to it. It was very simple. There was one rule regarding how well this was fitting the desired outcome that drove this solution. But if you didn't know that and you just see this final generation with 5 cells that are behaving in a way that defy any explanation, you might think this was quite a complex solution to be proposing to your problem. And I think in cancer biology, we're quite often confusing the complexity of the solution with the complexity of the process that led to it. And that is something that probably as statisticians sorry, so people aren't statisticians, but as statisticians we're well placed to try and say something useful. So I'd like to just stop there and thank colleagues now who various sites within the ICGC prostate study, colleagues in St Andrews who've made me most welcome since I've arrived here, and colleagues in Cambridge who I worked with for a long time. Of course, we also have to acknowledge the input of all the patients who've donated the material to these studies, the ICGC and some similar ones, in the knowledge that there will probably be no benefit to them arising from these studies, but purely for the benefit of future generations of patients. And finally, I'd like to acknowledge the various people that have funded me doing this work over the past few years. Thank you.
Deputy Principal, ladies and gentlemen, it's now my very pleasant task to deliver a vote of thanks to Professor Lynch for the excellent and insightful lecture he's just delivered. It's a real pleasure to see how ideas can travel between the rigid subject areas we sometimes define to allow exciting progress to be made on a disease that I'm sure has had an effect on too many of us who are sitting here in this room tonight. As we've seen, cancer is a multifaceted condition that requires a highly complex understanding in order to begin to develop successful treatments. Professor Lynch has shown us that it's only through acting in a global and concerted way with teams of scientists and clinicians working together that we can begin to address these requirements. It's fascinating to see how the Human Genome Project that addressed the whole human organism can be used to enable a battle at the level of single cells and individual genes. It's also fascinating to see how the very volume of information we seem to be drowning in these days can be turned into the route to develop and enhance our understanding through the application of the smart research tools developed by Professor Lynch and his colleagues. I reflect there's a quote from a now banned anti-cancer documentary from the 1970s that claims, "Cancer is a biological, not a statistical problem." I think that Professor Lynch has shown us tonight that it's only by understanding the statistical problem that we can begin to fully understand the biological conundrum that is cancer. And now it's my pleasure to invite you all to join us in Lower College Hall, where things that cause and cure cancer are sometimes confused. For example, there are some studies out there which suggest that alcohol may help to lower your risk of kidney cancer. So, with that knowledge in hand, I'd like to invite you all to join us for some medicinal beverages in Lower College Hall to reduce our collective risk of kidney cancer. Finally, I would like to thank Professor Lynch once again for his lecture tonight, and I wish him every success for his research in the future.